Well, it seems like The Walking Dead is one step closer to walking. That's right. Biological researchers have created dead zombie cells in the lab, and it turns out that they can actually outperform living cells. The team from Sandia National Laboratories at the University of New Mexico have innovated the technique where mammalian cells are coated with silica to form replicas, and these replicas work better than the originals. So joining us to discuss and explain all of this, we've got Jason Townsend, postdoctoral fellow at the University of New Mexico and one of the researchers in this study. Jason, hello. Thanks for joining us.
Hello. Thank you for inviting me.
Tell me, why did you want to create zombie cells? Don't we have enough zombie fear-mongering, scary movies out there?
The question why is something that comes up the very first time anybody learns about this, and there's a whole lot of reasons for that. But I have to jump back for a second to start. When you introduced it, you mentioned that the zombie cells can outperform biological cells or mammalian cells, and in some contexts that's true. But I think that part of the reason we did this, answering the why, you do have to fully realize the benefit, that the fact that we exist here and we're talking today, the function that all our different cells have, the actual different structural diversity. On the material science perspective, it's extremely difficult to recreate something like that. And so what we've done is essentially, as you mentioned, coated the cells in silica. It's something you commonly find in sand or glass. And what this does is it gives it structural support. And when we first tried this, we were simply looking at to see whether we could actually, coat or preserve what part of the cell, how much of the structure could we maintain. And what was phenomenal was that we found that down to the molecular level, even at the minor groove of DNA, we've replicated the cell. And in terms of its benefits and what you're saying, how it can outperform mammalian cells, live mammalian cells, that's due to the fact that this structural support it provides resistance to high temperatures, to dehydration, things that normal mammalian cells would not withstand. So the why we're still working on, there's a lot of different reasons, and they all have to do with all the different things you can normally do with material science, combined with the fantastic things that these cells can do normally by themselves.
So the why comes after we've already done all the work and realized that something like this can exist. But I mean, how revolutionary is this? How might this really change healthcare or in whatever way in which you choose to use this in the future?
Right. That's a great question, and it does come back to the why. We really were astounded to see that we could do it in the first place. We're working on the why, but in terms of healthcare, you could think of preserving, whether it be the different things that cells create, so the different protein structures that we could use for medicines and whatnot, and it would stabilize them so they could sit on the shelf. So you can imagine things where you need to stabilize the biological structure, where they may have to sit on a shelf for years. They may be in extreme temperatures in the desert, where I am right now in Albuquerque. You need that kind of stabilization. So this could help to stabilize the type of biological material and its function, so that it could be used under circumstances normally it would not be used. On the other side, one of the things that came up was on the energy side, that cells are extremely good, in terms of their catalysis or their enzyme function. And so combining the actual hierarchy of the structure and the function of the cell and preserving that may have very, very diverse functions. We're just beginning to explore the why and the functions right now.